This video is the continuation of the previous video, Nervous System Part 1. If you haven't watched it, kindly watch it and come back so that it will be more easier for you to have a continuation over the videos. Okay, now let's get into the video. In this video, first we will discuss about the functional classification. In previous video, we stopped with discussing anatomical classification of neurons. Now we will continue with the functional classification of neurons. We functionally classify neurons into sensory neurons and motor neurons. We said it in the name itself that we are going to classify them based on their function. Okay? Now, imagine a scenario. Based on the scenario, you will easily understand both the neurons, sensory and motor, and what are their work. Okay? Imagine a child is touching an iron box. What the child will immediately do? He will take his hand away from the iron box immediately, right? This is the scenario. Well, now let's get a bit deep into it. The impulse received by the child is taken to the central nervous system by the sensory neurons, and the command given by our central nervous system to the muscle receive the sensation is taken through the motor neurons. Okay, that's it. Now, let's go deep into these topics. The sensory neurons are further classified into primary, secondary, and tertiary neurons. It is very easy to understand. They are just all based on the location of their cell bodies. Okay. In these sensory neurons, whose cell bodies are all located outside the central nervous system, are called as primary neurons. Those neurons whose cell bodies are located in the central nervous system are secondary neurons. And for those neurons whose cell bodies are located in the thalamus or tertiary neurons. Okay. In these three, in the primary neurons, there is one exception. The mesencephalic nucleus. You might not have heard about this word. Okay, now let's see a little bit about this. Before getting into the trigeminal nerve and the mesencephalic nucleus, let's see what is nucleus. The collection or aggregation of cell body in the central nervous system is called as nucleus. In the peripheral nervous system, it is called as ganglia. Okay, that's it. So now we got to know what is nucleus. It is a collection of cell body in the central nervous system. The fifth cranial nerve, the trigeminal nerve, has three sensory nuclei and one motor nuclei. One of the sensory nuclei is this mesencephalic nucleus. This is present in the central nervous system. With the definition of primary, secondary, and tertiary neurons, I mean sensory neurons, we will think that mesencephalic nucleus is a secondary sensory neuron, but it is not. It is a primary sensory neuron, but it is located in the CNS. Okay, so this is the one and only exception in primary sensory neurons. Have this in mind, right? Now let's move on to motor neurons. In motor neurons, there are furthermore classification. First, we will see the classification in somatic nervous system, and then we will see in autonomic nervous system. Okay? In somatic nervous system, they are classified as upper motor neuron and lower motor neuron. Mainly based on their cell body location, we have classified them as upper motor neuron and lower motor neuron. Upper motor neurons, these neurons have their cell bodies in the motor area of brain. Okay, here there are many areas in brain. We will discuss about each and every area in upcoming videos. Now, get to know that in the motor area of the brain, the upper motor neuron cell bodies are present. They travel all along and they end up in the cranial nerves nucleus and in the anterior horn of the spinal cord, right? Up to this, they are all called as upper motor neuron. From the cranial nerves nuclei and from the anterior horn of the spinal cord, the neurons which were arising and traveling through are called as lower motor neurons. Okay, this picture I think is a good illustration. Look carefully. From the motor area of the brain, the upper motor neurons are arising. They are ending up in the somatic motor nuclei of brainstem, which is nothing but cranial nuclei. Okay, and in the anterior horns of the spinal cord. And from them, the neurons arising are called as lower motor neurons, and they end up in the skeletal muscles. Why skeletal muscles? Because somatic nervous system, previous video we have discussed what is somatic nervous system. They supply the voluntary muscles of our body, the skeletal muscles, right? That's it. Clear? Right? Now let's move on to autonomic nervous system, motor neuron classification. They are classified as preganglionic and postganglionic neurons. The name itself suggests preganglionic, before an autonomic ganglion, after an autonomic ganglion. Here also we classify them based on the location of the cell bodies. The cell bodies of preganglionic neurons are located in the central nervous system, and postganglionic neurons travel all along and reach the target tissue. Now, let's discuss about the fine structure of a typical neuron. A typical neuron will always consist of three principal components. I think most of us would have cleared our entrance exam or some exams before getting into this medical colleges, and definitely in every textbooks you would have seen these three organelles: cell body, dendrites, and axons in a neuron, right? Now let's see the fine structures in them. First, the cell body. The cell body majorly has a cytoplasm and a single nucleus with a prominent nucleolus. Right? Could you all appreciate these three things in this picture? It has cytoplasm, it has a single nucleus with a prominent nucleolus, right? And beyond this, we have two main characteristic features in a cell body to be appreciated. Whenever people ask about cell body, you have to tell about these two main characteristics. First is Nissl granules, second is neurofibrils. What are these Nissl granules? They are nothing but rough endoplasmic reticulum. When you see the picture of a neuron, whenever you go into Google or YouTube and type something called as neuron, you see a lot of dotted structures in the neuron called as Nissl granules. Why there is a lot of rough endoplasmic reticulum? Look here also you could see rough endoplasmic reticulums. What is the need of that? We need neurotransmitters for the transmission of impulses in a neuron. For synaptic transmission, we need neurotransmitters. Okay, for their synthesis, we need a lot of enzymes, and these enzymes are produced by this rough endoplasmic reticulum. Okay, so there is a lot of Nissl granules present. Then, what are neurofibrils? They are nothing but microfilaments, microtubules, and all the other cell organelles in the cell body. Okay, now could you all see the neurotubules visible here? Yes? Okay. These neurotubules are made up of a protein called as tubulin protein. We would have studied this even in our state board or NCRT books in 12th standard, right? These are necessary for the transport of large molecules across the neuron in both direction, in this direction and also in this direction. In either direction, when there is a need for transport of large molecules, we need neurotubules. And before going into the discussion of dendrites, I would like to tell you one thing that when there is cytoplasm, when there is nucleus, when we see a black color border covering the cell body, covering the axon, I have to discuss about that, which is nothing but the plasma membrane, which is the outer covering of a neuron. When the plasma membrane covers the cell body, this enlarged portion of a neuron, which is called a cell body, is covered by a plasma membrane. Then that plasma membrane is called as plasma lemma. When this extended single extended portion of a cell body called as axon is covered by a plasma membrane, it is called as axolemma. That's it. This is the minor difference between plasma lemma and axolemma. Okay. Now let's move on to dendrites. You could clearly see they are very short projections from the cell body. Okay. And they also have minute spine-like projections from them called as dendritic spine or dendritic gemmules. Okay, note this for an info. One important law you have to know in this dendrites is law of forward conduction or law of dynamic polarity. The word itself says forward conduction, dynamic polarity. The dendrites always conduct the impulses towards the cell body, okay, in this direction, towards the cell body, and never conducts impulses away from cell body. This never happens, and that's why we have given the law as law of forward conduction or law of dynamic polarity, right? Now let's discuss about axons. Axons are single extended portion of the cell body, whereas dendrites are many in number. Axons will not give many branches, but sometimes they give off branch called as collateral axon, whereas dendrites are always branched. Could you see? Right? Now, axons arise from cone-shaped portion of the cell body called as axon hillock. Could you see this cone-shaped portion of the cell body? Right? And there is a part in this axon called as initial segment of the axon. What is this initial segment? 50 to 100 millimeter of distance of axon from the axon hillock is called as initial segment of axon. Why is it so important? Why am I highlighting it here? An important MCQ. Note it. It is the place where there is the origination of axon potential takes place. The axon potentials originates in the initial segment of axon. Okay, and as I have already said, axons are covered by plasma membrane called as axon lemma. And now axons end up giving a lot of branches called as telodendria. And these telodendria end up in a bulb-like structure called as terminal buttons or presynaptic terminals. And these terminal buttons or presynaptic terminals contain neurotransmitters which are very important for transmission of impulses. Okay, and we have come to the conclusion of the video. Thank you for watching this video. If you like this video, please like, share, and subscribe the video.